I just want to start by acknowledging that this project has really taken on pretty significant global partners to make it happen, particularly because of COVID. We've really relied on our partners in China to do a lot more work than we thought we would need them to do. So I'm grateful for that. But we do continue to also work with the University of Rennes, where I did my Fulbright with my colleagues there, who are experts in polyploid plant genomics. Moment. Okay, so I took the opportunity to go back to the IPCC report that was released last fall and remind myself and all of us the basic take home from that. We have the highest CO2 concentrations in 2 million years, the fastest rates of sea level rise in at least 3,000 years. Arctic sea ice and glaciers are retreating. And of course, we've heard a lot about that in this series, as well as the extreme heat, heavy rainfall, drought, fire weather, and ocean acidification and warming events that have happened. So my work is in this context, really interested in how are the organisms going to be responding to these various stresses. And my work has always been interested in the mechanisms of response to novel or changing environments, but with the increasing pace of global change, it's become even more relevant. So one thing that's pretty interesting about this Make Our Planet Great Again group is all of the work that people are doing on the physical processes and the chemistry and the global climate. And it's not always so obvious how climate change affects plant communities. I apologize in advance to Jim that I don't have one of his wonderful examples here, but this was one of my favorite early examples when I started thinking about the impacts of climate change. And that was the understanding that there was new habitat basically being revealed in the Antarctic areas as these glaciers were melting and ice was sloughing off. So this was a report that was made available by the National Geographic Association about new species of grass and pearlworts that were discovered in Antarctica as new habitat was being revealed. And then of course, there's this story of the tree mortality in the western part of the North American continent, where we heard about drought and temperature interaction driving mortality in trees. So in both of these cases, we have some clear examples of how climate change affects plant communities. And in these types of stories, the basic focus is on how is it that plants are able to respond to novel environments. So this is a classic question in plant biology. We have lots of decades of researchers who've been interested in this idea. And this is one of my favorite examples because these are sunflower species that have diversified within very near habitats. On the left, you have the active dune habitat in the western deserts, and then right nearby, you have the stable desert floor. And in this case, we have one species that lives on the active dunes and one species that lives on the more stable desert floor. So these environments seem pretty similar to us, but for these plants, they're different enough that these are now different species. And Lauren Wiesberg has done quite a lot of work to understand how it is that they've diversified even when they're right next door to each other. So the focus of these types of studies and my work is really on phenotypic variation. So in layman's terms, this is trait variation. How do the aspects of an organism vary within a species, within populations? And then we think about how this phenotypic variation affects the way an organism is able to interact with its environment, basically its ecology. So this includes interactions with the non-living environment, like salinity and water and nutrients, as well as interactions with the biotic environment. So other animals, plants, all the organisms that are out there fall under the umbrella of ecological interactions, which are all facilitated by phenotypic variation. And through these interactions, we basically have the action of natural selection because there's variation in the population. You have some individuals do better than others, and those are the ones that will be more successful. This natural selection then can change the genetic makeup of these populations, which affects, of course, gene expression, and that affects ultimately the phenotype. So it's basically through iterations of this cycle that we get differences in populations. And this has been the classic foundation of understanding how biology works in organisms across the board: bacteria, fungi, plants, animals. So now, in the context of climate change, we might wonder how does this alter our basic understanding of these biological processes? And from my perspective, I think about how climate change alters natural selection, which of course means that there are certain genetic variants that might survive better than others. And that means the species or the populations will quickly evolve potentially. They may shift their range through some type of behavior or some type of migration, or they could be lost in general, ultimately resulting in extinction. Another option that we think about in my group is that the new environment might favor invasive species, in which case we have another complexity to think about when we're thinking about organismal response to climate change. So the invasive species aspect of global change is really an interesting problem for us as biologists, because it's basically presenting a paradox to us. An invasive species should have no genetic diversity because it's coming from an area where it's in a normal population, there's normal levels of diversity, but there are only a few individuals that are being transported. So in this hypothetical example, which isn't that hypothetical because it's very relevant for my Japanese knotweed work, you have a few individuals that were selected in Japan or somewhere else in Asia, and they're brought to Europe, and then eventually a few individuals make it to the US. And you can imagine that only a few individuals are going to be able to survive that trip. And therefore, you're going to have what we call a bottleneck. In fact, for the Japanese knotweed, we have really good evidence from several different labs, including my lab, that we have no genetic variation within the Japanese knotweed population. So just to orient you, in case you haven't thought about how genetic fingerprinting works, each one of these columns here is representing an individual. These individuals were collected in different places around the world. For example, you have studies down here from London, France, Germany, the US, Czech Republic, another study from Germany and Switzerland. Our studies were done in the US. Oliver Bosdorf's studies were done in Germany and Switzerland. Across all of these different studies and individuals collected, we've found that there is no variation, which is indicated by these bands here in the rows, that every single individual in this gel, and there are something like 20 or 30 of them, have the same genetic makeup. So this was the early evidence for Japanese knotweed. There are several other invasive plants that have this type of evidence where there's very limited genetic variation, or none in some cases. So in that case, if natural selection is favoring invasive species, somehow it's doing it without genetic variation. And this again is a conundrum for us, which is why we have thought a lot about how do you get genetic variation without, or how do you get phenotypic variation without genetic variation? And that's why we think about epigenetics. So in this case, the study of modifications to DNA that can affect gene expression and therefore the function of the genetic code, but not because there's a change in DNA sequence. So these mechanisms basically regulate the genotype, and they include mechanisms like DNA methylation, histone modification, small RNAs. Also, the location within the cellular matrix can influence which parts of the DNA are expressed. So these are all things that we haven't thought about traditionally in terms of understanding how you can get heritable differences between individuals within a population without any sequence variation. So just to give you a little more information about how does this work, this is a simplification of the concept of DNA methylation here. So the gene promoter region is the little bit of DNA that's right next to the gene that actually tells the machinery in the cell to transcribe the DNA. So what's happening is that in a normal expressed gene here at the top, you have a promoter region that has some bit of sequence like CCGG. So these are the base pairs that you think of when you think of DNA makeup. So you have A, G's, C's, and T's. C's and G's, which are cytosines and guanines, are particularly important when you're thinking about methylation. And that's because you can have a methyl group stuck onto these cytosines. And what happens in that case is the gene can be silenced. So to think about it in an intuitive way, what's happening is that this promoter region is where the transcription machinery sits on the DNA and runs down the DNA to make RNA. If you stick something on the promoter region here, in this case, a methyl group, you're basically interfering with that process. So that's a simplified summary of what can happen with DNA methylation. And so as you can see, it doesn't alter the DNA sequence. It can happen very rapidly. It doesn't require a mutation. Mutation rates are typically thought to be very, very low. And this methylation reprogramming can be altered by some kind of shock to the system. That can be either something like a climate stress or other type of environmental stress, or events like hybridization where you have genomes coming together during reproduction, for example. And it's important to think about this as a heritable phenomenon because it's something that can change the way the DNA is making proteins eventually without having a change in the DNA methylation, and it can be inherited in the next generation. So this is where some of the real interest is in ecology and evolution, because you can get change in a population across lots of individuals much more rapidly than waiting for DNA mutation to happen. Okay, so one really nice example that was discovered early on in this field of what we call now ecological epigenetics. It wasn't even ecological at the time, was the study of this cute little plant called Linaria vulgaris. So this plant has a natural bilateral symmetry here in the top figure. So this thing has, if you split it down the middle, it has mirror images of each other instead of a radial symmetry like a daisy would have. But sometimes in nature, you can find a more radial symmetry like this lower figure of the Linaria. And researchers in the mid-90s thought that this was probably due to a mutation, a natural mutation that just segregates in the wild. So you can find both of these types. The radial symmetry is not nearly as common. But when they went to look for that mutation, they discovered it was in fact a change in methylation pattern that drove this difference in phenotype. So this has always been a very exciting example because it was one of the early pieces of molecular evidence that change in DNA methylation can actually affect phenotype. So coming back to our diagram here, the way that we think about how change is happening in natural populations, how populations might be able to respond to some kind of climate change event, we think that epigenetic effects could be an important component. And we published a paper in 2008 with my host here in Germany that talked about how you can have a much more rapid response and a heritable rapid change in populations based on epigenetic variation. So that's part of the research program that we're pursuing here. And I'll talk a little bit about it when I get to the data we've collected. So back to knotweed. This is the native Japanese knotweed habitat. It is a primary colonizer in Japan. So this is Mount Fuji, in fact. And on the left there, you can see seedlings of this wonderful little plant that are establishing. You don't see seedlings so often in the invasive range because it also grows by rhizome, and it's very, very happy to grow by rhizomes. Also, it's limited in the invasive range because one of the species is only female. It can't pollinate itself. So again, this invasive species paradox, we think there's no genetic diversity in Japanese knotweed, but there's very high trait diversity. And it's a real conundrum for us why the plant looks fairly normal in the native habitat. So this is a photo of one of our colleagues in China. The understory plant here is the Japanese knotweed, not the bamboo that's really tall and huge, but this little baby plant that he's measuring. And we know very well the history of introduction because there was one researcher, one botanical fanatic that lived in Japan at the time in the early 1800s and brought Japanese knotweed with him to Leiden in the 1830s, and then sent it to London and all over Europe, in fact, where it then made its way to New York. So there are a couple of interesting stories about how knotweed made its way across the globe, but the main source of the introduction is from von Siebold, who was really interested in making it a horticultural plant. And so you can see the photos here that I showed in the Strasbourg meeting, that despite the fact that it's a pretty normal plant in China, it is super aggressive in Europe and the US. It's much bigger, at least anecdotally, we've noticed that it's much bigger. Now we have the data to show more about the native populations. And we don't understand how this can happen with a plant that's limited by genetic variation. Right, so I showed a version of this slide in the Paris talk a couple of years ago when we were first talking about setting up the knotweed experiments. But basically, I'm just going to walk you through the types of experiments that we think are important for understanding whether genetic or non-genetic or both are contributing to phenotypic variation. Also, our expectations. So here we have native populations on the top. And if you can see these different colors, we expect that individuals 1, 2, and 3 here are made up of different genotypes. So they have, for example, this gray gene that's the same, but the blue gene is only present in 1 and 2, whereas it's a green version in 3. And they're also different in this last chunk of genome. These little triangles are representing the methylation marks, which also are differing across the 3 individuals. So native populations, we think, should have a normal level of fairly high diversity among individuals, and methylation is going to be variable as well. Invasive populations, on the other hand, we think should have exactly the same genotype, but could vary in methylation patterns. So this is something you would expect to find for sure if you were looking at populations in the field. What we really want to know is how much of those differences in methylation pattern are maintained when you bring them into a common environment. The reason this is important is because these populations are experiencing a lot of different environmental conditions, and these methylation patterns should be responsive to them because they're associated with gene expression. We know that genes are going to be expressed differently depending on which environment you're in as you turn on different parts of your metabolism to deal with high sun or low water or high nutrients or whatever. An organism is going to change its methylation patterns. But if those patterns that are induced in the field are then also inherited in the next generation in a common environment, they could have important implications for the organism, despite the fact there's no genetic diversity. So this is the basic construct of how we go about looking for the importance of non-genetic inheritance, including DNA methylation and other types of epigenetic mechanisms. So our populations in New York, I'm going to tell you about some of the data that we had already coming into the MOPCA project, because we worked on these populations in New York fairly extensively while I was a postdoctoral fellow with Massimo Pigliucci, which incidentally is where I met Oliver Bostorff, who's my host here at the University of Tübingen. So we have quite a nice long history together working on this system. So in Long Island, this seemed like a really great opportunity to explore these ideas because the plant is living in lots of different types of habitats. It's normally found along roadsides and railways all over the invasive range, but we also discovered that it had somehow established on the beaches of Long Island as well as the salt marshes. And since my previous work had been in native salt marsh communities, I knew quite a bit about the physiology of some of the plants that live in these coastal habitats where they're exposed to salt and low nutrients usually and drought. So I was really interested in the differences in physiology that would be important for these different types of populations. So we did quite a few experiments exploring the different populations along the beach of Long Island. This is Suffolk County, Long Island, the farthest eastern part of Long Island. The beach populations, the marsh populations, and the roadside populations, which are interspersed kind of haphazardly here. We did a couple of different studies to explore the differences among these plants. One was a greenhouse salt tolerance study where we basically took rhizomes from the different habitats and the different populations within each habitat. We cut them up into pieces and we grew them under no salt or salt conditions. We picked salt because it seems like it should be a pretty important stressor for living in salt marshes and even on beaches. And what we got was pretty surprising because we found in the no salt treatment here, what's happening in these graphics, I should orient you. On the x-axis, we have the no salt treatment and the salt treatment. On the y-axis, we have a trait that's called succulence. Succulence is basically how much water the plant is holding in its leaves, which is thought to be an important trait for dealing with drought and salt. Because salt is sort of a different version of drought, preventing the plant from taking up salt. So we measure succulence by weighing the amount of water that's in a certain leaf area. So we weigh both the water and the leaf area of the plant to get this measure. And what we found is that in the no salt treatments, the plants pretty much perform the same. They don't seem to mind. But when you put salt on there, you get a lot of differences depending on which rhizome you're looking at. So these lines connecting the no salt to the salt, in each case, this is replicates of a single rhizome that we took out of the field. So this panel on the left are all individuals that we collected in a marsh site. And the panel on the right are 2 individuals from the roadside site. So this is a bit of a confusing result because there's no consistency in the response to salt, but there is variation among individuals in the response to salt, even though we've grown them in the greenhouse and they should be the same genotype. So we used our molecular markers to genotype these plants and discovered that they were all the same AFLP genotype. And in theory, they should be doing exactly the same thing in response to salt if they're the same genotype. So this was a bit of a perplexing result. Even more perplexing, well, in the same vein, actually, was that we took plants from each of these habitat types, beach, marsh, and roadside habitat types, you can see on the x-axis here, and we planted them back into that same habitat type and measured response of the growth, basically. So here you have final height on the left, leaf number in the middle, and leaf area on the right. And in every case, we have some examples of plants, particularly the plants that came from the beach, that are doing worse in this case than the plants from the other 2 habitats. Again, these are plants that should be more or less the same genetically. So at some point, they're getting a programming. They can see you. Oh, this is my daughter.
Hi.
Honey, please.
Okay, bye.
Thank you. So somehow they're getting programming of the habitat that they came from and holding on to a response that we don't know what it actually means for the performance of the plant. Right. So we used these markers, which basically can give you an indication of methylation differences among individuals, among populations. And we found that, in fact, there were differences in methylation in the plants from the different habitats. So here on the y-axis, we have proportion methylation This is an example of 7 or 8 different loci here on the x-axis, and you can see that the beach habitat plants have higher methylation in some of these loci, and the salt marsh plants, for example, have lower methylation in these other loci. And this is after growing the plants in the greenhouse. So they're holding on to their differences in methylation. I've listed the limitations of this approach, though, because we were using molecular markers, which basically just give you an indication of differences in chunks of DNA. It doesn't tell you exactly which genes are involved or what the function is of those genes. Part of the reason is because we're working with a plant that has a really big genome. We don't have a reference genome for it. And even the model plants where we do have references, they're not very well annotated to dissect this type of response. So in essence, we had a lot of exciting information to think that something's going on with this plant that's allowing it to live in these different habitats, despite the fact that it should be the same genotype. But we really needed more genomics resources to be able to dissect those ideas. So that was the framework going into the MAPGA. And basically, we argued that we had a lot of really great evidence to think that this was an opportunity to look at response of a given individual across a variety of climates and contexts. So this is the design that we proposed. We had 3 transects that were 2,000 kilometers each. And we also were happy to collect six populations in Japan, thanks to a colleague of our Chinese collaborators. Within each of these transects, these 3 main transects, we collected 250 individuals along a transect at 50 locations. And for each individual, we collected trait information in the field, as well as collecting plant leaf samples so that we could look at the genomics, and we collected soil, and we downloaded the climate data from BioClim. So the basic concept that we were working with here is trying to understand what are the components that contribute to organismal response, and then also what are the molecular level mechanisms that underlie those responses. So here you have basically a type of path that we were exploring, where we start with climate and biotic environment. Those environmental factors affect the growth and maybe the defense of the plants. So defense in this case are chemicals that are going to deter herbivores or pathogens. Those both affect each other, of course, but also contribute to performance of the plants. And this performance drives the impact that the knotweed invasion has on the communities. So we measured lots of stuff for each of these main components, including canopy cover and soil nutrients and herbivory for the environmental factors. We had leaf measures for understanding defense. Of course, we measured height and stem diameter and leaf area for the plants. We tried to capture some measure of performance by estimating the size of the knotweed populations, and we counted the density of stems and have measures of overall growth. And the community structure we captured with types of species that were in quadrats where we collected the plants. So I'm going to show you some of those data. I presented these briefly in Strasbourg, and a lot of this work has been done by Dr. Ramona Irimia, who's a postdoctoral fellow working on this project. And she has collated a lot of different approaches to analyzing this data. It's been quite an endeavor. Of course, it's a lot of data. But so far, we have some nice information about how the climate and biotic environments are different between the ranges, which is kind of surprising. I guess it's not that surprising when you think about it, but it was interesting to see that China fell out here on this ordination graph on the left side, separated from the European and North American populations. So there are a lot of potential ways to think about how climate's varying and which climate things could be important. But it is interesting that China is separating out from the two invasive ranges on this ordination. And as you may know, when you do these types of ordination analyses, the first axis explains the majority of the variation. So this includes things like the pH and magnesium of the soil, as well as the bioclimate variables, which I've listed here on the left, which are mostly related to temperature and precipitation. Right, so looking at the growth, defense, and performance of the plants, the graphics here show North America on the top in blue, and Europe in green, and China in red at the bottom. So you can see the knotweed density in the first figure is much lower in China than it is in the two invasive ranges. The stem height is not quite as tall as it is in the two invasive ranges. The leaf specific area is also less. So in all cases, we basically have smaller and fewer plants growing in the invasive range. We measured percent herbivory because one of the favorite ideas in invasive species biology is that enemies at home will be keeping plants down basically and making them behave normally, whereas when they get to an invasive range, those enemies aren't there anymore. So the plants can shift their resources instead of defending themselves against their enemies, they can shift their resources into more growth and performance. We found that there wasn't really a good indication that the herbivory was different in the three ranges. But what we did find, which was pretty interesting, is that the majority of the plants in China had some evidence of pathogen, whereas in Europe and North America, we had very low evidence of that. So this could be an important driver of the differences between the invasive and native ranges that we'll be following up on. So getting to the molecular level processes that contribute to these responses, we basically had three strategies: more precise genotype and epigenetic markers. And the rationale for using markers is that the genome of the plant is really large and intractable at this point. So it's about twice the size of the human genome, which requires a lot of resources and money, and it's complicated to dissect because it's also octoploid. So basically everything about the genome makes it pretty challenging to get really fine scale information about these individuals. We also planned to do gene expression studies because gene expression is a nice readout of genetic variation and genetic differences among individuals. And it's a phenotype, so it's closer to a functional level of what's happening in the organism. It's also more affordable because it's only the part of the genome that's being expressed. And you can have lots of copies of each gene in an octoploid plant that end up making just one product, so that you have basically data reduction when you're focusing on gene expression. At the same time, we thought it was important to develop a whole genome reference for this plant, because then we have much more information about the gene expression as well as the genotyping markers, because then we can map it back to a genome that we have more information about. So we are pursuing all three of these approaches. Number 2 and 3, I'm not going to talk about today because they're very much still in progress, led by our postdoctoral fellow, Armand Cavero-Day, working with Armel Salmon and Professor Malika Enouche at the University of Rennes. I will give you a little bit more information about the genotyping in knotweed, because I mentioned in the last meeting that we think there might be some unexpected results here. And basically what's happening with these unexpected results is that we so far have looked at a subset of our sampling. Our postdoc here, Dr. Isolde van Rimsdijk, has done 11 individuals of the Japanese knotweed that I've spent most of the talk talking about. One individual from each population spanning the full 2,000 kilometer gradient in Europe. She has discovered that there are 2,500 DNA sequence differences in these individuals. So I just want to remind you, this is kind of a shocking result for me, even to this day. We've had this result for several months now, but we expected these individuals to be exactly the same genome. So to get 2,500 differences in DNA sequence changes up the whole storyline quite a bit. At the same time, we also have this other species, which is a hybrid between our Japanese knotweed and a plant called the giant knotweed. So this is really widespread in Europe and also very invasive, and we have many of these in our collection. That hybrid species has 8,000 SNPs. We expected to have more genetic variation in the hybrid because obviously two genomes are coming together. The hybridization has happened several times in Europe, but we still only have we should only still have a few genotypes because it spreads pretty rapidly by rhizome. So here's another ordination for you to see what that looks like in reduced dimensional space. So in this case, we're using a principal components analysis to squash down all the variation in all those markers. You can see the Japanese knotweed, which is the 8X, looks like there's not as much variation here is basically what's being depicted in these ellipses. Whereas in the hybrid, you have much more variation among individuals, are the green guys here. So what's a little bit more disturbing, and I apologize that this graphic is challenging to interpret when you're not used to thinking about these data. But on the y-axis here, we have genetic distance, which is basically a pairwise comparison between two individuals. You compare two individuals to see how different they are from each other. And that's what each of these individual little dots are on this graph. So I'll start with the US and the EU knotweed, which is this 8X over here, because this is the surprising result that I mentioned before. Across the US and the EU, we find that pairwise comparisons are actually not giving us an answer that they're all 0 down here, which is to be expected because maybe there is some mutation, maybe there's some error. The technology is not 100% perfect, and the coverage that we have to get really precise measures is not high enough yet. So here you can see there are quite a bit of differences. It's not 0 for one, which would be a nice ideal result. There's maybe less variation among the hybrid, but that's partly a problem of sampling because we have fewer of them. And then here are the Chinese populations. So this is the other take home from this graphic is that the Chinese populations, we expected to have huge amounts of diversity. And in fact, they don't really look that different from the US and the EU at this point. This green line here is the line that our colleague Mark Schmidt has calculated is probably the limit for detecting a single genotype. So given the error, given the technology limitations, all these guys above the green line, we really think they're not the same genotype anymore. So that's the take home of our story so far. So we've made this big story about how one of the world's most invasive plants is a single clone. It's living in all these really diverse habitats. It's pretty phenomenal. It is a female plant, so it makes sense that it can't produce lots of variant offspring. But now we wonder, is it really just one clone? So that's, I'm going to leave you with that question because it's actually one of the biggest burning questions in our research program right now. But I also want to just recap that we have a lot of challenges that we're facing for understanding mechanisms of response to these really complex environmental conditions. The number one is that most knowledge of genome function has been acquired in model species and in controlled environments of the lab or the greenhouse. The lab is really where most of the work has been done. Sometimes researchers have ventured into the greenhouse, but that's a little uncontrolled for some molecular biologists. We need to measure response of complex genomes like the one that we're working on and complex environments, because that's where biology really happens. Also, dissecting genetic from epigenetic mechanisms is challenging even in the most well-understood species. So that's a whole other lecture that we could go into. There's a lot of discussion about this, a lot of debate about whether it could be important or not. But the reality is we just don't have enough data to really know. Then there's this new idea that we hadn't thought about for the first 10 years of doing this research, and that's the importance of mutation. We had assumed that it must be pretty minimal, but now we have evidence that it could be pretty important in this system. So I'm excited to see what happens in the next year or 2 or 10 with these questions. But I just want to remind everyone that our ultimate goal here for describing the system, explaining the associations between traits and climate variables, soil, bioclimate variables, is to eventually be able to predict what's going to happen in the future. We also have this enduring question of how much does this system reveal for other systems? So these are big questions, big pursuits that we hope to shed light on in the coming years. And with that, I'm just going to thank all of the people, many of the people, probably not all, but I do appreciate all of the support, especially my family who moved here for the MOPCA project. You saw Dorothy wandering around, and my son John is also downstairs. My host here in Tübingen is Oliver Bosdorf, a longtime friend and colleague. We've had really great times working together. We've had lots of students and postdocs before and during this project, as well as really great colleagues from all over the world. This is a snapshot of one of our MOPCA Zoom calls. I know you all are familiar with these now because that's how we have been doing science for the last 2 years. But it's a really great group, and it's growing all the time. And I appreciate that I've had the chance to be part of this. And that's it.
Okay, thanks a lot, Christina. I'm sure there are many questions, so you can ask them as normal, either by pulling them into the chat or into the question and answer tool, or by just raising your hand. If there is none yet, then I might ask the first, which is maybe a little bit unrelated to the talk, but can you actually talk a little bit about the impact of the knotweed on in its invasive range? So because invasion is of course a negative connotation, so
yes,
does it have negative impacts?
It definitely does. It's one of the worst invasives on the planet. There's a really interesting article that was written a couple of years ago for Slate magazine. The reporter asked me for my ideas about knotweed for that article, and he summarizes the impacts because they're fairly devastating, particularly in the UK, because there the plant has invaded a lot of personal property, and it's actually caused devaluation of the property for a lot of people, and it's a pretty massive problem. There's also been tons of money spent on trying to get rid of it. It interferes with things like the railways, and it destroys concrete, and it invades into buildings. It's a pretty impressive plant in that respect. It seems to be able to destroy things, and also the ecological impact is pretty massive. So when we look in the field where knotweed is living, there's nothing else there in terms of the plant communities. It just kind of wipes everything out and takes over. It grows tall and dense, and nothing can live under it.
Is there any human use of it? Any potential use?
Because it's so massive, it's actually been a target for biofuel development. And I think there are biofuel companies here in Germany that are growing it for that purpose. I'm not sure exactly how that's working, but it grows fast and it puts on a lot of biomass. So it does make sense for that type of application, that if you're looking for a plant that's going to be super productive, super fast, it fits the bill for sure.
Mm-hmm. Okay, James Clark raised his hand. James, please.
Hey, Christina, very nice talk. and unexpected results, I guess. So I am familiar with this plant, and going into this, you now have a very different view of the genetic structure and how that could play into this whole problem. And I guess I wonder, even though you're not all the way there yet, how do they go about managing for this now? And given the insights you have so far, how does that change the way you might think about going about managing Japanese knotweed?
Yeah, that's a really important question, and one that we talk about often with our colleague Bernd Blasi, because he's really interested in trying to develop the biocontrol for it. So I think what ends up happening at the end of those conversations is usually trying to control it a little bit, but not thinking that you're ever going to get rid of it. So with some of the biocontrol agents that have been seemingly successful, at least to some degree, they don't seem to be able to completely kill the plant. But if we can figure out a way to knock it back a little bit so that other things can also live there, that might be the next goal. I don't know if that
I guess as a follow-up, do you think that it will get to the point to be more like it is in China, and it'll just the community trophic interactions will adapt to the point where it's less of an issue?
Yeah, I wonder about that. I mean, I don't know how long it would take for that to happen. We don't have any good examples of that kind of storyline. I guess because it's sort of an unprecedented time in terms of translocation of organisms around the planet. It's been a fairly short period of time that we've had such massive translocations. So I think eventually that could happen, but what we find in the European range is that we don't seem to have much of anything that's impacting the biology of the plant once it gets a foothold. You know, we don't see the pathogen impacts, we don't see the herbivory. There's a weevil that invaded our greenhouse collection, which did knock it back a little bit, but didn't totally kill it. But that was an isolated, like with the plants isolated in pots. So yeah, I'm not sure if it's going to ever be at a point where it's in balance. Let's see.
Thank you.
Thank you.
Okay, other questions? If not, I'll ask another one. I mean, if we think about the invasions, would you then say if such things can happen so massively that there are also huge niches open in the environment? Or, I mean, sometimes we always think that, I think we have a natural feeling that the nature is how it is and that all the species that we are in our surrounding are best adapted, but maybe it's not like that. Or what is your
Yeah, so I think that's a misconception, really, that I'm not sure where it comes from, but I get it. I mean, I think that it's there. The problem is, and the climate change scenario brings it into stark focus, right? Because the environment that the community's adapted to is not the environment they're going to see in the coming years. So that's one of the concepts that we think about, is that maybe just the disturbance of so much change opens up the opportunity for other things to come in. The real question is, why is it that some things are really able to take advantage of that? Because they don't have the fuel. We normally think of genetic diversity as the fuel for that type of response. These guys have giant genomes. So another version of genetic diversity for them could be that they've got 8 copies of every gene, and you know, you only need 2 as a diploid organism. You function well with 2. So then you've got all these extra copies that can do additional stuff. So that's one of the favorite hypotheses of why polyploid organisms can do well. But then there's the flip side of, they have all that genome that they have to replicate and hold on to. And, you know, it's an interesting field, because we know through evolutionary time, there have been whole genome duplications that have been really important for diversification. And we know that across evolutionary time, certain copies of genes will just get shunted off. So the organism doesn't actually have to hold on to all those copies. And it might be more economical to just get rid of some of the genes, some of the copies of genes once you have so many. So it's a pretty fascinating field to think about how the genome can really accommodate a lot of different problems. But why is it so able in the invasive and the natives seem to be just kind of sucking at it? I don't know the answer to that. It's just part of the changing landscape, I guess.
Okay, maybe related to that, if I understand it correctly, the polyploidy might also be important in the methylation story, because if you have more genes, then you actually can turn more on or off.
That's right.
And actually then you maybe have, I mean, turning something on or off is not really the new function, right?
Right.
Is that correct?
That's right.
How fast can the methylation be turned on and off? Is that a random process, or is the plant actually can it be? Is the plant controlling it to some extent, or what is actually doing that?
Well, we don't know a lot about how it's actually done, other than we know enzymes that sort of catalyze the reaction, and there's an interaction between RNA and DNA methylation. So there's a complex that's somehow registering where the methylation needs to happen in the genome, and it's pretty tricky in terms of really understanding what's driving it. We think that it's not completely driven by some other part of the genome directing where the methylation is going to be happening, but we don't know how much of that is possible.
Mm-hmm.
We don't have enough data and enough circumstances to really be able to tease those things apart.
Okay, thanks. Then we have a question by Chantal Reed. Are the pathogens in the native range specific? Given that there is a higher proportion of pathogens on knotweed in its natural range, are there any attempts to look at introducing one of these pathogens in the invaded range?
Yeah, that's a great question. So we don't know anything about the pathogens yet. We've just discovered this pattern in the field. And in fact, it wasn't identifying pathogens, it was more identifying the damage that they made on the leaves. So this is a line of inquiry that we're going to be developing, we hope. It's going to really rely on the Chinese lab picking it up and trying to identify some of those pathogens. So we don't know yet anything about that. We are also interested in the microbiome more generally, because we know that the microbiome can be pathogenic or facilitative. So we're hoping that we can pursue some of those lines of inquiry.
What do you think generally of the idea of introducing pathogens?
That scares me, because you just never know what's going to happen when I don't even know how you would test for that, but they have protocols for testing for biocontrol. I don't know how far it extends into the microbial world, but it might be a thing we could think about. The problem, of course, is that the pathogens could attack other stuff once you bring it into a different territory, and you don't know exactly how it's going to respond once you put it outside.
Yeah, there were some stories from islands where the rats were introduced, and then the snakes were co-introduced to kill the rats, and the snakes killed all the birds on the islands, right?
Right, exactly. You just escalate the problem. I think Nuria has her hand up.
Oh, sorry. I think she took it down again.
Yeah, thanks,
Sorry.
Christina. No, that's okay, Rainer, that's okay. And thank you, Christina, for your talk. Very nice. And also, I need to let you know that also I share unexpected results. That is very challenging, but sometimes it's very difficult, no? to understand what is going on. And also, when you work in natural population and in the field and so on, so you need time to understand the system. And okay, this was a small comment. And I have I don't know, an idea that you have about the genetic structure, about the different populations, that that you have show us, let's say, the different genetic structure from the different sites, the study sites. And I don't know if it could be interesting to see over time. I don't know if you have the possibility to go to the museums or, you know, to go back to the time.
Yeah.
And to see if you might have, or if you might find some, you know, some signals over time. I don't know if this could be, you know, a good idea. And also I don't know if you want to comment on that.
Oh yeah, so that's a really great observation. And in fact, one of the postdocs in our group has a project that she just got funded through the Marie Curie Fellowship Program to do that. So she's going to be collecting samples from the different herbaria around Europe and trying to figure out how they've changed over time. So it's really a nice opportunity because we know pretty much exactly when it came to Europe. And we have a nice documentation of Philip van Siebold, who was in the Netherlands. And he basically just sent it out to all of his friends and distributed it around Europe, and also was selling it as an ornamental plant. So her project will focus on using herbarium samples and doing the genetics of those samples to try to see if they're the same, if when we can maybe reconstruct if there were mutations coming up and how they diverged maybe over the landscape. I mean, it's a pretty exciting idea if we could recreate how it moved across Europe and maybe changed over time in different places, and maybe actually get at a little bit of this question of how important is mutation, how common is mutation. Too, yeah.
Okay. And the other question it's a little bit, you know, connected with what you were saying with Rainer about transplants. I don't like a lot of transplanting and post-transplanting, but I don't know if you have thought about, you know, the invasive populations to transplant back to the native origin.
We did do that, exactly. That's part of our project. So it was very complicated because sending live plant material to China was challenging. But yeah, we managed to send samples from our entire garden. So we have almost 250 samples here from the 50 different populations. And we took pieces of rhizome and sent them back to China. And then also from the US, it was a little more complicated. We could only send 50 samples, but we sent everything to the China garden, and we plan to do some experiments where they're all in the same place so that we can see how do the plants respond to the herbivores there in China, and what happens with their chemistry and their gene expression and their epigenetic mechanisms. Can we get an idea of the differences between what the native plants do and what the invasive plants do? So yeah, we hope to do that. Of course, it was really, really slowed down because of COVID. It took a long time to get the collections. It took a long time to get the collections from here to there. And then they were delayed because China had a lot of restrictions. But we have a garden there now. So eventually we should have some pretty interesting experiments.
Okay, good. Thanks.
Yeah,
Thanks, Christina.
thank you.